I want to just draw our attention to the poll results. Interestingly, we have about 56 or so percent of our audience is in the translational early stage clinical trials, followed by about 33% or so preclinical only. So interesting as we move on through our discussion. I want to talk a little bit about planning an end-to-end donor pool management strategy. So starting with high quality donor material is obviously critical for allogeneic cell therapy drug manufacturing. And this requires laying the groundwork for a high touch end-to-end donor pool management strategy. Koki, I'd like to start with you. What are the components of, say, best practices for effective donor management?
Thanks for the question. I think you're going to see a lot of similarities between what Lauren and I do. I think we are having different platforms, but the same challenges. So these are great questions. When we think about donors, I think he touched upon it. It's so important for people to realize the difference between autologous, which is a patient-derived immune system, which is pretty beaten up, typically an older patient. So when we think about the promise of what we do, it is the healthy donor premise. It's higher immune fitness. And toward that, we think about at Atara building out a robust materials management team, as well as strategic planning functions. And really what that means is to your point, how do we identify those healthy donors and how do we bring them into our strategic inventory for our production process? I think that what goes lockstep with that is a really productive relationship with our CDMOs and CROs that help us find those healthy donors. And then I think the last thing, and maybe this will keep coming up as a theme, seeing the poll results, is you always have to have this vision for how do you implement your supply chain now that's phase appropriate, but then you can look forward so it's pivotal ready for all of those compliance concerns that are globally pretty thorough. So in other words, when we're in the phase 1 space, sometimes you're RUO and you're barely phase 1, you just want to get efficacy, but you really have to have that long-term vision for when you go pivotal or commercialized, it's a whole new ballgame. And maybe Lauren can speak to that too. Thank you.
Yeah, absolutely. Thank you, Koki. Lauren, I want to give you the floor too, and perhaps even, like Koki said, building on what he offered, the components of best practices for effective donor management. What are your thoughts?
Yeah, certainly agree with everything that Koki had to say. I think the theme that you're going to sense as we move through every topic is preparation for the next phase, right? And I think one of the things that we realized is we thought we had a very robust donor pool in terms of size. And as soon as you start seeing some success, as soon as you start adding more products to the pipeline, as soon as you want to start making more CMC changes, even the things that you want to upgrade later in the process require more donors to evaluate, right? So it's not just what you're doing in the clinic and how many clinical patients you're trying to get through the process. It's also the support of your future CMC activities, right? So everything goes in tandem. There's a lot of demands that get placed on your donor pool. I think, looking back on my experiences in the autologous space, the larger organizations, they put into place some patient operations groups, which obviously in the autologous space makes all the sense in the world, right? You've got to manage the patients, you've got to get them in for their leukapheresis. They're obviously very sick already. That's a big lift. And then secondly, they need to have visibility to what's going on in the supply chain and when they can expect to have their product back. I think very similar in the allogeneic space, we need to start looking at donor operations as its own unique portion of the supply chain. And one of the things that we found is you also have to recognize the human attributes, right? You're already dealing with a supply chain that puts us in a very uncomfortable place. Most of us in the pharmaceutical world like to have super tight controls on all of our incoming raw materials. And the most important thing we receive comes out of a human being the day before we get it, right? So it's a very different space, and we have to make sure that we recognize the human elements of what goes on in the donor world as well. So one of the things that we found is that it's very important to build a predictable recall strategy so that if you go through a screening process with the donor and you find that their material is compatible with your process, how do you engage them quickly enough so that they don't lose interest in the process? What's the right pace and methodology for reaching out to them? And then secondly, you also have to keep very close tabs on what their health is, right? And there has to be a deferral process for people that get sick. And you also have to have a reinstatement process for when they're ready to get back into your donor pool. So it's truly and very literally a living, breathing pool. And it takes a lot of management and a lot of oversight to make sure you don't find yourself coming up short on the most important starting material that you have in allogeneic cell therapies.
Good, good. Okay. I want to talk a little bit more about, you talked about certainly the importance of the inbound materials and keeping tabs on that. If we could talk a little bit about that a little bit more, maybe explain some of the considerations for maximizing the consistency on those inbound materials, which are so crucial to the entire process.
Yeah, so, a couple of things, at least from our standpoint, we found that certain donors, even if they meet all of the initial screening criteria, either don't do a great job through our manufacturing process, don't expand as well as other donors, or even if they do, they may or may not control tumor in our in vivo efficacy tests. So we've got a couple of control points where we can evaluate how donors perform, and we make decisions about where each donor should be, whether that's a donor that we want to use for GMP operations, whether it's something we want to be only using for research purposes, or whether it's just a donor that's not very conducive to getting any information or effective material out of. And then I think the other thing that you brought up is consistency of the materials. And one of the big decisions that each organization has to make is, are you going to use fresh inbound material or are you going to use frozen inbound material? We've used both in different programs. And while the operations folks have a strong preference for frozen material because it doesn't create a rush, the clock isn't running in terms of how quickly you need to get to it, there's also some extra considerations when you start talking about frozen material, in that essentially that's an extra process that you're adding, that's an extra freeze thaw, you're putting some more stress on the cells. And then secondly, how do you make sure that that freezing process is consistent at each center, at each point in your network before you receive it, right? So that is a lot of extra steps to put out onto your providers. Secondly, your other option is to have it come inbound fresh and then freeze it yourself. But again, that brings in all of the headaches associated with fresh material. So there's no perfect solution, but whichever solution you select, you definitely need to put the work into characterizing how you're going to get the material, how you're going to evaluate each donor, and what the purpose of each donor is going to be.
Yeah, thank you, Lauren. That all makes perfect sense. And Koki, I wanted to give you the opportunity to also weigh in here on perhaps your and Atera's take on the considerations need to maximize the consistency for inbound materials. What are your thoughts?
Yeah, this is one of the biggest problems, I think, that maybe many people didn't anticipate, to Lauren's point. Your product is as good as the donors that you can identify. And part of this process for us at Atara, as a commercial GMP product, it's educated us internally about how rigorous you have to be. You have to define these critical quality attributes. And Lauren addressed this, to address and assess all the variables in PBMC collection. So there's the donor identification, there's how the PBMCs are collected, how they're shipped, how you then formalize receipt and cryopreservation. None of this is trivial, requires tremendous attention to detail for regulatory requirements in the EMA, as well as here in the US, in addition to anticipating GMP quality standards. And it's a process in the sense where there's screening, there's collection, and then obviously there's all the follow-up post-collection screening of the donors. So this is really a big operation, and I think it's essential part of being successful in our field.
Sure, absolutely. Koki, you mentioned the regulatory requirements just briefly there for donor management and inbound materials, and I want to talk about that just a little bit more. What are the regulatory requirements of donor management? How have you and your team handled it, you know, maybe some best practices, if you could talk us through that a little bit.
So one of the cornerstones of what was built out before I came to Atara is we have a relationship with Be the Match. So it's a large registry of healthy donors that are pre-screened in the sense that we know the HLA alleles, which is central to our technology. So we have a very thorough system for proper informed consent. And then there's regulations, right, for pre-screening, for eligibility. So there's you know, screening against adventitious agents. We want to have all of the blood cell counts within normal ranges, healthy donors. And then obviously there's the post-collection aspect again, and that speaks to the regulations for, again, maintaining that what we're putting into a patient from a healthy donor is safe for the patient. And on top of that, there's obviously HIPAA regulations. And then, you know, even on top of that, there's GMP quality concerns for how did you collect these cells? These are important process intermediates. Can you track this throughout the process? So for me, when I look at it, to your question, it's really regulatory, it's quality, and then there's obviously federal regs for de-identification of donors.